All right, everyone. It is exactly 1 o'clock, so it's time for this particular session. So, on behalf of the Lewis Stokes Midwest Regional Center of Excellence, we welcome you to today's session, the oral presentation, Top 25 Abstracts, Group Number 2. So, my name is Dr. Monica Frazier. I'm the director of the Southwest Georgia LSMP Alliance, and I am at Columbus State University in Columbus, Georgia. Just some housekeeping items before we get started. So before I introduce each of the speakers, I'm going to provide some general housekeeping items. So just so you know, this session is recorded and it will be made available for viewing through the WOVA app by next Wednesday. And so we ask you to provide feedback for today's session in the post-session survey. And so we'll make sure we have that survey there in the chat for you so that you can enter your comments or what have you. If you do have any questions for the presenter, I ask that you please use the discussion board to post your question at any time, just so that the speakers can present uninterrupted. And then as soon as their session is over and they're done speaking, I will then open up for questions for the presenter. So this particular session is scheduled to take an hour, so we need to make sure we stay within the time. But if we run out of time for the Q&A at the end of the session, we will encourage you again to use that discussion board to continue the conversation or to reach out to the presenter via the WOVA app. So at this particular time, I will go ahead and introduce the very first speaker, or I should say speakers. So if we have a dynamic duo from the University of Guam. So if you will, go ahead and start your presentation.
Thank you, Dr. Frazier. Let me share my screen. There we go. Can see it?
Yes, we can see it.
Let me start. Ali and Mogathin, my name is Matawan Choi, and I attend the University of Guam studying political science and communications. I am from the Pacific Island region of Micronesia, currently at the island of Guam, 3 a.m. in the morning. So I'm glad that I could make it. I culturally identify as Palauan Yapese.
Hello everyone, my name is Junita Jetli and I attend the University of Hawaii at Hilo. My major is Administration of Justice with a minor in Anthropology, and I am from the island of Saipan. I culturally identify as a Chamorro, and I apologize for all the rooster sounds.
And our project is STEM Journeys of Pacific Islander Students across the Islands of Opportunity Alliance. Iowa LSMP is a 5-year project running from 2018 to the year 2023. Over here is a picture that we took from our kickoff meeting back in January 2019. Our project goals is to better understand student beliefs towards their success in the STEM fields. We measure this by asking them about their community values, their responsibilities, their interest in careers in STEM, and their sense of social belonging to the scientific community. Our research questions ask, 1, what are student experiences and beliefs towards STEM? And 2, how do students understand their motivations and success in their STEM programs? Our research design is qualitative. We have students serving as research apprentices, such as Janita and myself. Together, we are culturally responsive. We engage and reflect on research experiences. We support with our cultural insight and responsiveness to the project. And overall, we become mentors to understand our environment and our identities within the Pacific Island region. Our research methods this past year was to hold focus group interviews. This consisted of 12 interviews with the 10 campuses aligned with our Iowa LSMP. We held Zoom calls, had informed consent given out so we could obtain the data. We digitally recorded these Zoom calls, these focus group interviews, so that we could later transpose them into transcriptions, but also add them to our coding scheme. And here's a little graph at the end where we wanted to share the STEM degrees that we found through our interviews. And in general, we found that across the Pacific, students are interested in various fields in STEM, especially marine science, marine biology, natural resource management, and environmental marine science. And these are the degrees we found throughout the Pacific Island campuses. Altogether, all student participants from their diverse backgrounds want to build stronger STEM programs, but also give back to their community. I would now like to hand it over to my partner, Janita, who will speak on behalf of what we found through our research.
So there are 9 Indigenous Pacific Islander communities that our research impacts, and they are students of Hawaiian, Chamorro, Samoan, Palauan, Marshallese, Pohnpeian, Cochran, Chuukese, and Yapese descent. The geographic area is larger than the continental US, and as you look to the right, you can see the geographical region of the 9 Indigenous Pacific Islander communities. What we have found in our research includes the major themes of motivations and goals to pursue STEM. So in our first subtheme of commitment to community, a student from the College of Marshall Islands voiced that the community is what motivated them to complete their marine science degree. For a second subtheme of financial strains on academic journeys, a student from the Ho'olu program at Chaminade University stated that they are experiencing a hard transition with working and attending school at the same time. And for our third subtheme of research experiences, a student in the Lafau program at American Samoa discovered that the reefs are an important part of their Samoan culture. The preliminary results show that the motivations to pursue a STEM degree are commitment to their island communities, financial strains, as well as with the guidance of their LSAMP research experiences. The next steps of our research includes the coding of our previous interviews, as well as conducting new interviews for the next academic year, which will capture the experiences of students returning to the LSAMP program. Lastly, we would like to thank our faculty members, Joseph Gens, Tobias Irish, Monique Story, as well as student apprentices, Tasha Kukui Akana, and Eusebio Ort for their assistances. On behalf of my team and I, we would like to thank you for joining us today.
Thank you. Dr. Fraser, do I stop sharing the screen? You're on mute.
I'm so sorry. I was going to say, either way, it's up to you on that one. Okay. If you want to take it down, or because you may have to reshare if there are questions about the poster or the graph. So at this point in time, we will take questions from the audience. Are there any questions for these young ladies? All right, so I will ask one question. So how was the experience of collaborating and working with someone at an institution not at your own? How was that? What was that like?
It was fun. So many emails that you have to answer because we could not just call each other directly. And having most of our classes, because of the pandemic, transition to online, having Zoom as the platform for our communication, but also email correspondence, really helped us bond together because we could not meet and travel to meet each other. Yes, that's what I thought. What do you think, Janita?
I thought it was amazing getting to know Mertuan and just having to figure out the time difference with everything, as well as scheduling. But because we are from the same region, we were able to bond in a lot of things. Because Saipan is basically Guam's sister, so we're like right next to each other, and we're just like, oh, you know this person, so we just like formed the connection, and it was just amazing.
Very good. Well, I will say you definitely learned a great lesson in collaborations. One thing about STEM is that collaboration is key, and it's very important to the advancement of your projects and whatever it is that you're doing. So you guys have learned a very valuable lesson in terms of how to collaborate, how to do it effectively, even in the midst of different time zones. And I do appreciate you all being up at 3 a.m. your way. It's nice and dark over there. So thank you so much for being up and for presenting your research with us. All right,
Thank
so at
you.
this time we'll move on to the second presenter. So at this particular point, we will go to North Central College. And just to reiterate, the session is being recorded. And so it will be made available for viewing by next Wednesday in the WOVA app. And if you have any feedback about today's session, please complete the post survey session survey, and then make sure that you post any questions that you have for the presenters in the chat and in the discussion board. All right, so at this point, we will go to the second presenter.
Hello.
North Central College. All right.
Okay. Is my mic okay? Sorry.
Yes.
Nice. Okay. Hello, my name is Julie, and today we're presenting on the effect of manganese on the recolonization of vegetation by plastic. What are mycorrhizal fungi? Of fungi, they live in about 80 to 90% of all plants, and they form a mutualistic relationship with the plants. They provide protection from pathogens, drought resistance, and weed control, but their main function is nutrient intake. The fungi will provide the plants with nutrients such as phosphorus and manganese, and in return, the plant would give the fungi carbon. Manganese is an important micronutrient that plants need to make photosynthesis. Our question is, how do different levels of manganese affect plant and fungi performance? And our hypothesis was that as manganese concentration increases, so will plant size and fungal proliferation. We thought this because more manganese would mean more photosynthesis, and with more photosynthesis, that means more. This experiment, we separated our big blue sim into two separate groups, one with fungi and then one without. Each week they were given different concentration of manganese ranging from 0 parts per million to 256 parts per million. After they were harvested, we weighed them on a scale, and the leaves were taken to Oregon National Lab to be examined by OH and HCl, and then we dye them using trypan glue. The trypan glue allowed us to be able to quantify it under a microscope, and here's what the fungi look like under a microscope. So you can see little hyphae coming out of the vesicles. So we found that as root colonization increases, the size of the plant also increases, as seen by the positive slope in our chart. We also found that as manganese concentration increases, the size of the plant actually decreases, and we can see that there is a little negative slope going on. And we found that plants with fungi grew a lot better than those without. So the green bars represent, the dark green bars represent the plants that had fungi, and overall they usually a lot taller than those without. And the plants grew a lot better above ground than the ones below ground. So below the zero is how much they were underground, and then above the zero is how much they were above ground. Then we lastly look at how manganese concentration affected root colonization, and we found that there wasn't really any interaction between them. A lot of the bars are about the same height, and there's a lot of overlap in the error bars. Although visually there was a slight difference. So these were the photos that were taken at Oregon National Lab. And you can see that the plants that did have fungi in them, there was a lot less manganese present in them than in the plants that didn't have the fungi. So like more red, greenish color represents more manganese was present in the spot. So our hypothesis was that as manganese concentration increased, so will plant size and fungal concentration. However, we found that manganese actually decreased plant size, and root colonization did improve plant growth, and the plants that had fungi grew a lot better than those without. The fungi themselves were not affected by the manganese pollution. It can lead to oxidative stress, otherwise known as manganese toxicity. Under manganese toxicity, the plant will create a free radical oxygen, and the free radical oxygen photosynthesis. However, as seen in the previous slide, because the fungi allow less manganese to be reach the leaf, we believe that it's possible that the fungi protected the plant from the high level of manganese.
Uh, Julie, I think I kind of lost you at the end.
Oh,
Your 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 sound was going in and out. So were you done with the poster?
yeah.
Oh, okay. I'm sorry. Okay. At this point, I will ask the participants in the audience if there are any questions for Julie. Any questions? One question in the chat is which AMF you used?
So we actually use it's our basket mycorrhizal fungi, and we didn't come
I think we lost you again, Julie.
in the wild. Yeah, my mic's there you go. In the wild, there's like various types of mycorrhizal fungi, and we wanted to keep it as closely at the specific type.
Okay. Any other questions? The Maria says thank you for answering that question. All right. If there are no other questions, thank you again, Julie, for a great job and for your work that you have, the research that you've done. So thank you so much for coming and presenting.
Thank you.
Okay, we've had a couple of more to join after Julie's presentation, so I'll just reiterate that this session is being recorded and that it will be available through the WOVA app by next Wednesday. And so if you have feedback about today's session, we ask you to please complete the post-session survey. And then, of course, for the presenters, you have about 5 minutes to present, and then we will allow people to ask questions at the conclusion of your presentation. And we ask that you submit those questions in the discussion board. So at this point, we will go to our third presenter. So we're going to the great state of Arizona. So Danielle, are you ready?
Yep, I'm ready. Can you guys hear me?
I think you're on mute, maybe.
Uh,
Okay, we can hear you now.
oh, okay.
Great.
All right. And then you guys can see my slides.
Yes.
Perfect. Awesome. Hi, everyone. My name is Danielle Pace, and I'm a senior undergraduate researcher at Arizona State University in the Brownell Biology Education Research Lab. Today, I'll be discussing my research concerning student support and accommodations for students with disabilities during COVID-19 pandemic. So COVID-19 has proven a challenge for both universities and students nationwide, as the emergency transition to remote learning served as a catalyst for dramatic instruction changes and a redefinition of what a college learning experience is. This new modality of online education has transformed what constitutes a class session from what happens in a physical classroom into a series of videos, online discussions, and online assignments. So our previous spring 2020 work revealed that many STEM students with disabilities were not properly accommodated after the transition. For this particular study I'm discussing today, we decided to collect data 1 year later in spring 2021 after universities had a significant time to prepare online course formats. So we had 4 main research questions for this study. The first one being, to what extent did science students with disabilities who reported new challenges in online learning report being properly accommodated? And what challenges were not accommodated for? Another was, to what extent did receiving new accommodations affect science students' perceptions of whether they were properly accommodated? So what new accommodations did the students receive? What ideas do students have about online science learning environments
can be improved for students with disabilities. And our final one was, what specific accommodations do students recognize as important for improving their learning experience? In this study, we surveyed students with disabilities registered with the Disability Resource Center at a single large enrollment research intensive university about their experience receiving accommodations in their online courses. We asked open and close-ended questions about their experiences with online learning and used logistic regression and open coding to analyze our data. So, did students with disabilities feel adequately accommodated online? Of the 114 students registered with the DRC, only about 30% reported not experiencing new challenges related to their disability in regard to online learning. And of those 30%, only 24% reported that they were not being properly accommodated. We can see in the side-by-side comparison, about 61% of students who reported experiencing new challenges also mainly reported that they were not properly accommodated. Students who experienced new challenges were significantly more likely to report not being properly accommodated. So this table depicts some of the top new challenges of online learning reported by students, such as video proctoring software, accessing distraction-free testing, extra time on computer, instructor not recognizing accommodations, instructor forgetting accommodations, additional test time, and accessing video lectures. In our second finding, we found that receiving new accommodations did not affect student perceptions of whether they were properly accommodated in the online learning environment. This table shows a regression of novel challenges and new accommodations. It shows that students receiving new accommodations did not predict whether a student felt like they were properly accommodated. And so when discussing the improvement of online science education, students identified both what additional accommodations were needed and how delivering accommodations needed to be improved. And so below are two quotes from two students. From Lauren, there needs to be updates in professor knowledge about extending test times when courses are online. And then for Kyle, I have been asked to provide warnings in advance when my disability will cause me to need an extension. But with my disability, I'm not able to predict when I need help. Overall, we found that many students with disabilities were not being properly accommodated, and there was a lack of standardization in the accommodations. One major issue we noted was the lack of instructor accountability, and this seemed to be a systematic problem. Through our study, we developed the following recommendations: increasing DRC trainings, requiring instructor training to support students with disabilities, monitoring instructor compliance, creating communication pathways between instructors and DRCs, encouraging institutions to share resources, and more research and development for online accommodations. To end the presentation, I would like to thank both WASO and the Arizona State School of Life Sciences and my amazing mentors, Dr. Sarah Brownell, Dr. Caitlin Cooper, and Logan Jin, as well as the Biology Education Research Lab and the Cooper Biology Education Research Lab. Thank you so much.
Thank you so much for that presentation. That was very awesome. I do have one question in the chat for you. And the question is, what method of research did you use for your research, and what are the challenges faced?
So we used an a survey with closed-ended and open-ended questions to analyze students' experiences. We based it on our 2020 study. So some of the questions were based on our interview study of how these students experience challenges, initial challenges with that online transition. Sorry, I forgot about the other question. What was the other question?
The other question was, and what were the challenges faced?
Some challenges faced were just understanding and coding the responses. We used open coding, so we had other researchers help identify themes that were throughout these student responses in the survey, in the second survey, and just making sure that we were understanding what these students were saying.
Okay, and your second question is, over what time frame was the survey conducted?
So our survey for 2020, it was conducted throughout the spring semester. So this spring semester, through January through about March. So students had an adequate time to respond, and instructors had an adequate time to send out the surveys as well.
All right. Any other questions? Let me see. I got one more thing in the chat here. It says, if I follow the design correctly, these are all students who were students in the prior year. Do you think challenges experienced by entering freshmen into the program are the same, or are there new challenges?
If I understand your question correctly, are you asking like if freshmen were also like asked these surveys and submitted these surveys?
I think they're asking if, so if the design that you use, these were all current students who were there the year before, so they were returning students. And so the question is, do you think the challenges experienced by entering freshmen would be the same as the returning student, or would they be new challenges as a entering freshman?
Yeah, so this survey is a bit different from the previous survey. So the 2020 study was sent to those students and those freshmen, but this is a 2021 survey. So this would send to even the freshmen entering the life sciences and the science courses. So this is a new group of students. Overall, we had about 2,000 student responses, including freshmen, sophomores, seniors. Yes, yeah. So it included new freshmen as well. But I'd love to also do a follow-up study kind of examining like the current freshmen and what challenges they have now that they're back in person, and seeing if there are more challenges associated with that transition.
All right, thank you so much. Very interesting study and very much needed work for sure for this particular population. So thank you so much. All right, so we will move on to, we'll stay in Arizona, except now we'll have a different student. We'll have Kaylee this time. Again, just a reminder, the session is being recorded and will be available to you on next Wednesday. And please provide feedback about this particular session. So Kaylee, you ready?
Yes, thank you, Dr.
All
Frazier.
right.
Hi, everybody. Can everybody hear me pretty well? I just want to make sure.
Yes.
Okay, great. I'll get started. Hi, my name is Kaylee Paddock, and I'm a junior here at the U of A studying physiology. And today I would like to present to you my poster entitled Noise-Induced Sleep Disruption Reduces Orexin, Orexin-1 Receptor, and Heme-Regulated Inhibitor mRNA in Female Rats. First, I'll begin with the background. To begin, societal noise pollution causes sleep disruption and increases risk for metabolic diseases that include obesity, type 2 diabetes, and cardiovascular disease. As we know, sleep is required for several physiological functions that go further than curing acute sleepiness, and this may include weight maintenance and normal metabolism. Orexin is a neuropeptide in the brain, and estrogen is a circulating hormone that is abundant in females. Both bind to specific receptors to modulate the sleep-wake cycle and regulate body weight. Heme-regulated inhibitor, or HRI, is one of 4 kinases that is important because it regulates protein synthesis and the response to stress. Currently, it is unclear whether changes in orexin, estrogen, and HRI signaling are related to weight gain caused by noise-induced sleep disruption. The purpose of this study was to determine if gene expression for the estrogen-1 receptor, or ER1, orexin-1 receptor, or OX1R, and HRI were affected by noise-induced sleep disruption in a rodent model of sleep disruption-induced obesity. I hypothesized that noise-induced sleep disruption will increase weight gain and food intake, and also reduce the gene expression of ER1, orexin, OX1R, and HRI mRNA in female rats. Next, I would like to direct your attention to Figure 1. This outlines both the study design and the procedures. First, 3-month-old Sprague Dawley rats were used for this study. They were given a period of 7 days to acclimate to the housing facility. Then the female rats were randomized by weight to 2 treatment groups. Each group contained 7 to 8 rats per group. The first group was the control group. These rats slept ad libitum. they ate, slept, and moved more freely. Next is the treatment group, or the noise-induced sleep disruption group. This treatment group was exposed to pre-recorded environmental noise for 8 hours a day for 9 days during the light cycle. Because rats are nocturnal, they sleep during the day. Has been validated to cause sleep disruption by reducing both the duration and quality of sleep. During the study, both body weight and food intake were measured daily. After euthanasia, the hypothalamus was dissected and flash frozen. Next, I processed the hypothalamus. First, total RNA was extracted from the hypothalamus with a quick RNA mini prep plus kit. Then total RNA was measured on the nanodrop spectrophotometer. This measured both the purity and concentration of total RNA. Next, all samples were reverse transcribed to make complementary DNA or cDNA. Then gene expression for ER1, orexin, HRI, and the housekeeping gene GAPDH were measured in duplicate by qPCR. Finally, the data were analyzed by separate t-tests for each gene. Now I would like to direct your attention to figures 2 and 3. These figures outline the results. As seen in figure 2, exposure to environmental noise significantly increased both body weight and food intake. This is indicated by the star over the gray bars. The gray bars indicate that this is the treatment group that was exposed to pre-recorded environmental noise. In figure 3, panels A, B, and C show there was a significant reduction in orexin, OX1R, and HRI mRNA in rats that were exposed to pre-recorded environmental noise. In contrast, panel D, there was no significant difference in ER1 mRNA between the two groups. In conclusion, these data demonstrate that low hypothalamic orexin, OX1R, and HRI mRNA may contribute to both elevated weight gain observed previously during the noise-induced sleep disruption in female rats. First, I would just like to thank my amazing PI, Dr. Teskey. She was so supportive throughout this process. And I would like to also acknowledge that the funding for this research was supported by WASO, or Western Alliance to Expand Student Opportunities, the National Institutes of Health and Environmental Health Sciences, and the U.S. Department of Agriculture. Thank you very much.
Thank you very much. Very good. Are there any questions for Kaylee? Any questions from the audience? All right, so Kaylee, for the study itself and the noise, I know you said it was pre-recorded noise. So were these animals kept in a facility separate from the ones that did not have any noise, such that the ones that I guess did not experience and had a peaceful sleep were able to do so and not hear the noise on the other side? And how did you ensure that? Was it just simply a different room, or?
Yes, so in the facility, there were 2 separate rooms. The first room had the treatment group where they were exposed to pre-recorded environmental noise. And this room was insulated to ensure that both the noise stayed in and also did not come out. And the second room contained the rats that were ad libitum, or that they slept, ate, and moved more freely.
And at the conclusion, were you able to weigh the animals to see if you could kind of see, because how long was the duration of the study?
Yes, over the duration of the study, we found that both their weight and the food intake were increased in the rat population that experienced a pre-recorded environmental noise.
All right. Any questions? Any other questions for Kaylee? All right, Kaylee, so you just let us know, make sure you have nice, quiet places to sleep, and we don't have to worry about so much of the weight gain. I'm sorry, we do have a question. How many days or hours was the duration?
Yes, so the rats were exposed to the noise-induced sleep disruption for 8 hours a day for 9 days during the light phase. Because rats are nocturnal, they move around and scurry during the night, and they sleep during the day. We expose them to the noise during the day because that's normally when they sleep. So of course, that's how they were sleep deprived.
So the person with the question says, "Wow," because I'm with them. That is a long time to hear some noise,
Mhm.
Next question is there a direct effect of food on the mRNA levels, or is it the causation, or is the causation the other way? So did food have any impact on this mRNA level, or was it the opposite?
I am not too sure, but I do understand that the sleep deprivation itself reduced a lot of the gene expression in the orexin and the HRI gene expression, but it did not necessarily affect one of the other genes called ESR1.
You have another question. Are these results applicable to wild animals that are nocturnal?
That is a really interesting question. Currently, we only used a female rat model, so I am not too sure if that is applicable to other wild animals, but that would be really interesting to look into.
Let me see, we may have one. Okay. And were you able to measure the difference in effect on REM and non-REM?
No, I'm not too sure about that portion.
Okay. Yes, you didn't look at see if their eyes was blinking or something for the REM, huh? All right. Any other questions? All right. Well, good job. Thank you so much, Kaylee. We appreciate your work.
Yes, of course. Thank you, everybody.
All right. So we've had a few more people join in. This is great. Again, the session is being recorded and will be made available for viewing through the Nuova app by next Wednesday. Again, we do ask you to please provide feedback about today's session in the post-survey session. And if you have any questions for the presenters, please place them in the chat. So at this point, we will move to Indiana, to the IUPUI, and for our second to last presenter. We're doing pretty good on time, I think.
Can you hear me okay?
We can.
Okay, so I'll share my screen. Okay. Okay, so my name is Gina Ramirez. I'm an environmental science student at IUPUI, and I'll be presenting my research over reconstructing the history of anoxia and climate change in the Laguna Encantada and Chetumal, Quintero, Mexico. So lakes are sensitive to changes in temperature and oxygen. Dissolved oxygen is less soluble in warmer water, so anoxia, which is a lack of oxygen, is expected to become more widespread as Earth's surface temperatures increase in response to increased anthropogenic carbon dioxide. Some problems associated with anoxia can include harmful algal blooms, fish kills, decreased water quality, and these can have a detrimental effect on ecosystems, human health, and our drinking water. So the lake is called Laguna Encantada, and see if I can get my laser out. It is located near the coast on the Yucatan Peninsula in Chetumal, Mexico. Tropical lakes like Laguna Encantada tend to be less prone to mixing and are located in remote areas, which make them ideal for studying anoxia. Tropical lakes are also understudied compared to temperate zones. And there are also few reliable methods for determining how oxygen concentrations have changed with time in lakes. And originally developed for ocean sediments, the reactive iron proxy method uses the distribution of minerals that contain iron to determine oxygen content of water. So one aim of this study is to adapt the iron proxy method to develop a new methodology for tracking anoxia in lakes. In a warming world where anoxia can become more common, reconstructions of climate and anoxic conditions from biogeochemical alterations of lake sediment can provide information about how climate change can impact society in the coming century. So a 4.5 meter sediment core was collected from Laguna Encantada in 2019. Based on lithology, 3 distinct units were identified from the bottom to the top of the core. The oldest sediment is roughly estimated to be about 5,000 years old, and we're currently working with a colleague in Mexico to determine a sedimentation rate and adjust our results. 90 total wet mud samples were taken from the core for geochemical analysis. To determine total carbon and sulfur, we use the ultra carbon sulfur determinator. Total carbon and sulfur were determined from the CO2 and SO2 detected from combustion, and total inorganic carbon was determined from CO2 detected from acidification. Total organic carbon was calculated by subtracting total inorganic carbon from total carbon. To measure element concentrations of total iron, phosphorus, aluminum, and titanium, samples were dissolved through microwave digestion, diluted, and ran on the ICP-OES. So laminations of carbonates and organic matter at the bottom of the core in Unit 1, transition in Unit 2 into massive bedding in place by rapid deposition in Unit 3. Overall, the geochemical data changes align with the changes in lithology. So Unit 1 has higher organic carbon, sulfur, and phosphorus, which all decrease towards the younger sediment. Total iron and aluminum are also similar, so that can suggest that total iron concentrations are controlled by detrital input, since aluminum and titanium are known to be relatively immobile. Total organic carbon to total sulfur ratios also indicate that marine influence in the beginning of the core, with higher amounts of sulfur in Unit 1 transition into low sulfur, which can indicate freshwater conditions in Unit 3. There's also distinct separation in CS to total iron ratios between the 3 units as well. So our initial results show the story of a seawater influenced lake that transitioned into a freshwater lake over the course of 5,000 years. Reactive iron extractions of carbonates, oxides, magnetite, and pyrite are currently ongoing, and we anticipate the reactive iron to total iron ratios to provide a proxy of past oxygen conditions. Stable isotopes of carbon, nitrogen, and sulfur will also be analyzed, and that could correlate with Mayan settlement history in Yucatan. We also plan on aligning the geochemical results with history of sea level changes and modern climatic events in the Yucatan to observe possible correlations. Our main goal is to provide insight into future conditions and implications on our freshwater systems as climate change continues. Thank you.
All right, thank you. Great job. Any questions from the audience for Gina, right?
Janina, yeah.
Any questions for Janina? Any questions? All right, so I will say I saw your future goals there. So is this the goals that you have, are they in line with your future research in terms of, has it influenced you in terms of your career and what you would like to do?
Yeah, I mean, this project has definitely kind of guided me towards climate science. And so I am interested in a research career down the road in graduate school. So I do think I'll continue in this topic, in this research area, because I've just grown sentimental to my project. And I think I love the science of it too.
Awesome. You do have a compliment there that says, I honestly really appreciate the clean visuals. Great job.
Thank you.
There is a question. I am unfamiliar with the history of that site. How does the timing of the Mayan civilization occur at that specific location?
Yeah, so I did a little bit of background research, and I actually have an article in one of my references that details Mayan settlement history. If I'm not mistaken, they came around 3000 BC is when they settled, which translates, I don't know to how many thousand years, but I'm going to align the geochemical changes and then make a timeline of the Mayan settlement history to see if anything correlates, especially with their agriculture and just overall impact of the lake. So yeah, that is down the road after we get all of our data. Then that is one of the first steps I'm going to do for sure.
All right. Thank you so much. Any other questions? All right. Well, thank you so much for that presentation. We have one more to go. And we are going to Lewis University. But again, right before he gets started, and I'll give him time to pull things up, the session is being recorded and will be made available for viewing through the WOVA app by next Wednesday. If you would, please provide feedback about today's session in the post-session survey. And if you have any questions, please put those into the chat. So at this point, I will turn it over to our final speaker for group 2, Joshua at Lewis University.
Hello, can you hear me?
Yes, we can.
And my presentation is up?
It is.
All right, thank you. Hi, I'm Joshua Segura from Lewis University, and the study that I conducted was measuring odonate biodiversity through a comparative study of larval and adult life stages. So odonata are commonly referred to as their suborder as dragonflies and damselflies. We studied the larvae stage, which is spent in the aquatic habitat, and the adult stage, which is terrestrial. And in terms of studying these species, they serve as indicator species because understanding their niche behavior and biodiversity can help establish conservation plans. There are certain environmental factors that can affect the abundances, such as declines in habitat, increases in pollution, and land development. Therefore, attaining abundances is important to see if these factors are decreasing or increasing their abundances. Through this study, what we propose is if biodiversity estimates are influenced by life stage, therefore comparing adult and the larvae. And I predicted larval biodiversity would be greater since the likelihood of the egg maturing to adulthood is low. The two sites we investigated were the Morton Arboretum and Sagwa Environmental Learning Center, which is west of Chicago. For the methods, we utilized the Illinois Odonate Survey, which is done with a pollard walk, and you simply count the number of dragonflies and damselflies you see along a specific route. For collecting larvae samples, we extracted pond muck and other debris from bodies of water, such as streams and ponds. From there, we sorted out through this material and obtained the larvae and preserved them in ethanol or isopropyl alcohol. And then from there, we looked at them through a microscope to see what determining features would give their species, but we in turn identify them by family instead. The progression of the adult survey and just for the adult survey showed that abundances have a progression, therefore they can increase or decrease year by year, which is important because it shows that either sampling effort is not really being it shows that sampling effort can vary year by year, and it can also show that there are those environmental factors that can cause these increases or declines along the populations. And to compare the larvae and adults, overall, there were more adults compared to larvae, such as with table 1 showing 521 total adults and table 2 showing 1,207 total adults. Therefore, if you really want to look at biodiversity, the adults are a way to go. However, you run into either 3 scenarios where you have larvae and adults present, or you only have larvae present, but no adults present, such as shown with the Cordula gastridae family in table 1, or you have just adults present, but no larvae present, such as shown with the Cordulidae family, which represents that the adults are helpful and give insight towards attaining biodiversity, but there is a chance where adults might not be present. Therefore, basing off larvae is a way to kind of gain that biodiversity assessment of that site. For future directions, I think collecting more larvae samples is very critical, because if you make it more routine, you can kind of gain more numbers and abundances for larvae. which can give you more insight towards biodiversity. Along with this practice, narrowing down the species level is important because we only did family due to time constraints and the kind of practice that we need to gain from narrowing down the species level. Therefore, this can kind of correlate also to species dynamics, ecosystem dynamics that each species may react to, because some are more susceptible to environmental changes, others may be more tolerant. As shown with the adult survey, doing a year-to-year analysis is important too, because this can show how conducting those survey efforts are really strengthening that biodiversity assessment, because there might be a time where you really see some and you really don't see some the next time you survey. Therefore, having that progression can show that as well, and also with the change over time with the land development as well. These are my works cited, and I'd like to thank Nagin Almasi from the Sagwa Mental Learning Center for collaborating with us in this study, Rebecca Collins for issuing us the Forest Preserve District of Cook County permit, and Kurt Dreiselker and Cindy Crosby from the Morton Arboretum for collaborating with us in this study as well. And I'd like to thank the National Science Foundation through the LSMRCE program. All right, thank you for listening to my presentation.
All right, thank you so much, Joshua. Very good. Any questions from the audience for Joshua? Let's see what we have here to chat. How far do dragonflies fly? Is one question.
It depends from what I've known, like previous research about the certain articles that we looked at. There are some that are migratory, so some that may go from state to state, others may just reside in the area, which it can vary. Generally, it's based off species too.
And another question I overlooked, and it's kind of related to something I had as well, because you mentioned that there were more adults than larvae, and so I'm not familiar with the life cycle. But the question is it more difficult to locate the larvae or the adults?
Yeah, it's very difficult with the larvae because it's almost like a needle in a haystack. You're going to collect a lot of you can either collect a lot of debris and very little debris, and with a lot of debris you could have at maybe 45 larvae and just that pond muck that you obtain, or you can have one in all that pond muck that you obtain. Therefore, it varies by day. I believe that's why doing it more routinely, you can gain more pond muck, and it increases the likelihood of finding larvae.
Okay. How would you standardize results based on sampling efforts?
It's just gathering numbers. So we separate them by family, and from there we just kind of we would do like we could pool them. We could separate it by family if we were to show a progression. Is that what you were referring to?
I'm not sure, but we will see. I'll let them come back on that one, but I do
Okay.
have a follow-up to your answer on how far dragonflies fly. And the follow-up question is, will that affect your estimates of biodiversity for each site?
It can, because another primary component of collecting larvae is showing if that species is originating from that area. Because if you can have that family with there being 0 larvae and multiple adults. So that shows that the likelihood of an adult coming from a different area and habituating in that area shows that they did originate from a different area. So I think that kind of goes hand in hand with that as well. That'd be something that you could also test as well too.
Okay. I have two more. You said that, this is another comment, you said that comparing results would be based on how much effort you invested in collecting. You could standardize abundance by dividing abundance by time spent collecting or by number of dips to allow comparison from year to year.
Mhm.
That was a follow-up to the standardizing question.
Yeah,
And then you also have two other comments, which is awesome. Thank you and great job, Joshua.
all right, thank you.
All right, are there any other comments? All right, I think we kind of did this one. Oh, go ahead,
I think
Joshua.
Danielle has a question. Oh,
Okay,
Oh,
you can
sorry.
go ahead and
That was
ask
a
it.
congrats.
oh, I thought that was a hand raise. Sorry.
Let me see, we do have another thing in the chat. Okay, another great job, Joshua.
Thank you, Nadine.
All right. Well, thank you so much. That concludes the Group 2 Top 25 Oral Abstract session. Again, if you will, please go to the poll and enter in your comments about this particular session. I do appreciate your attentiveness for this particular session today. And I'd also like to say that, a reminder that this session is recorded and that it will be posted in the WOVA app by Wednesday of next week. And so again, if you will, please complete the post-session essay. And yes, congrats to all the students. Well done. Great job, speakers. And thank you very much, everyone. All right. There were great presentations. So thank you again. Just happy we were able to stay within time. So if there are other sessions you'd like to go to, you're free to go to that session. Thank you so much.